Our last speaker of the morning is Julie Millard. She got a bachelor degree from Amherst College, went to get a PhD in biochemistry from Brown University. So I've done all the IVs now. We're done, pretty much. Did a postdoc at the University of Washington, and was a Dreyfus Teaching and Research Fellow at the University of Richmond. She's now the Dorros Professor of Life Sciences in the Department of Chemistry, which is interesting, at Colby College. She is interested in small molecules interacting with DNAs, and I know something that you should be careful. Don't let her challenge you to a run or a race of whatever sort. I believe she's qualified for the Boston Marathon, so be careful in what you do.
Okay, thank you very much for the invitation to come here today. It's really an honor to speak to such a distinguished audience, and a little bit intimidating because I'm used to speaking mostly to non-majors this semester and a few students who want to go to med school, so I'm a little bit nervous, but hopefully it'll go okay. Okay, so I'm from Colby College. That's what it looks like right now. That picture was just on Facebook, so a lot different than the view outside. And I do study DNA and small molecules that interact with DNA. And sort of for the general audience, people know DNA is the molecule of inheritance. This was taken at the Smithsonian several years ago. My daughter is now a senior in high school looking at college herself. My son is in 8th grade. But DNA explains why siblings look alike and look somewhat like their parents and share a lot of properties, not only in people, but also in canines, so these are my dogs. Same mother, paternity is questionable, different litters, but they look a lot alike. And so we're all familiar with human chromosomes. This is a actual picture, a micrograph of a human chromosome. And as a biochemist, we like to look at molecules way down deep, the reductionist approach to biochemistry. And so DNA is packaged by wrapping it up in proteins. If you were to take the DNA from a cell and stretch it end to end, it's something like 2 meters long. And so it's condensed very tightly to be able to fit inside the cell, all the DNA that we have. And really, we're looking at it at this level, the double helix, and we've also done some work with packaging of proteins. And more recently, we're actually looking at DNA inside cells. So the DNA double helix is composed of base pairs held together by hydrogen bonds. We have AT base pairs and GC base pairs, and the agents that we're looking at are targeting guanine at this position right here, which is actually very helpful chemically, as I'll get to later in the talk. Small molecules that target DNA can have cell-killing potential, which is called cytotoxicity. That may seem like a bad thing because you may not want your cells to die. But the trick is, if you can direct this at cells that you don't want to have propagating in your body, that can lead to an anti-cancer drug. The molecules that we're looking at are DNA cross-linking agents. They bind covalently to each of those strands. The strands are normally held together by fairly weak hydrogen bonds, so the strands can come apart and the genetic material can be expressed and copied. But if you have a covalent linkage between the strands, you can no longer do this. So this can be deadly to a cell, and that's good if it's a cancer cell that this is happening to. The family of crosslinkers that I'm looking at are related to a molecule that was developed for gas warfare originally in World War I. So this is a very famous picture of people who are blinded and probably have lots of nasty skin lesions as well, because they have been subjected to mustard gas. Chemists were interested in developing more powerful compounds to use for gas warfare, and so systematically they started to vary. If they decided if they could put a nitrogen there in lieu of the sulfur, they could create a whole family of compounds that might be more effectively used for gas warfare. So this family of compounds are called the nitrogen mustards, with varying groups right here at this position. There were a couple of pathologists, a husband-wife team, who were doing autopsies on soldiers who had been gassed and found that the white cells had been devastated. And so it was actually proposed that perhaps these compounds could be used to treat things like leukemia or cancer of the white blood cells. And so nitrogen mustards were, you can't gas somebody, right? Because it leads to all these nasty lesions. So they developed these nitrogen mustards that could be water soluble, perhaps injected. They were used to treat lymphomas as well. And they found that, in fact, they were quite effective. And so nitrogen mustard with a methyl group right here became the first anti-cancer drug chemically synthesized and is still used. And there are other cousins with different R groups that are still widely used. So that compound, and this was the code name, they all had their code names, so HN2 with a methyl group in that position, that's called mechlorethamine, and it's often just called nitrogen mustard. And as I said, it's still used. I did a lot of work with nitrogen mustard as a postdoc at the University of Washington with Professor Paul Hopkins, determining sequence preferences targeted by that compound. When I got to Colby, I investigated this compound, which is a chemical cousin. It may not apparently look that way, but nitrogen mustards, when they are activated in the body in solution, they form an aziridinium. And so it's a three-membered ring with a couple carbons and a nitrogen. The chloride pops off. And so chemically that might behave similarly to an epoxide. Diapoxybutane has been linked to the high rate of cancer in butadiene workers because butadiene is metabolized in the liver in an attempt to water solubilize it to diapoxybutane and some other products. And kind of interestingly, it's also thought to be the active form of an anti-cancer drug used to treat ovarian cancer. So that drug is actually a pro-drug. It gets converted to diapoxybutane, and that's believed to be the active compound. So both molecules can be used to treat cancer, and we're interested in the mechanism by which that occurs. We've also looked at this molecule, which we consider to be a hybrid between the two. So it has that chloride arm, but it also has a single epoxide as well. So it also has two sites that can react. And this compound is one of those crosslinkers that we just heard about, an epoxide that's used in the synthetic polymer industry. And there are people who are exposed to it who have developed cancer at higher rates than would be expected. So it is a suspect carcinogen. So we've also been looking at this molecule as well. Can't be used to treat cancer. It's simply a carcinogen. So if you crosslink your DNA, that can also lead to cancer as well as cure cancer if you're targeting cancer cells. So the way DNA crosslinkers look, or the way they work, here's just a simple schematic. If we have DNA, two strands of DNA held together by weak hydrogen bonds, and we introduce a crosslinker, then the crosslinker first of all reacts with one strand, and then it may close to react with the other strand to form a crosslink. It doesn't have to. It could just react with solvent on the other arm and just end at the monoadduct. And the crosslink tends to be a product found in very small amounts. So maybe 1% of your total products, maybe 5% depending on the crosslinker, sometimes more. But these tend to be pretty infrequent events that you actually get the crosslink. The monoadduct would not interfere with replication. It may interfere with some other things, but the crosslink covalently locking the two strands together is really disastrous. Experimentally, you can separate out this crosslink even though it's present in relatively minor amounts. So say we have a population, we've treated the DNA with a crosslinker, we have some of the native, the unmodified, some monoadduct, and some crosslink. And the way we do this, or one way that you can do this, is use denaturing polyacrylamide gel electrophoresis, which separates compounds out by size. And the yellow strand, it denotes a radio label so that you can easily see your products on a gel. And so when you do this, the two strands separate because denaturing means that you're breaking up those hydrogen bonds. And all you see when you put your piece of X-ray film down or your phosphorymager is the radioactive strand. And so the native runs to some position on the gel. The monoadduct is slightly heavier because it has the compound attached to it, so it has a slightly lower mobility. And the crosslink has both strands, and so it's about twice the mass. And so you can easily visualize it about halfway between where you started and where the single strand would go. And so it's very easy to separate these out on a gel, cut out your crosslink with a razor blade, and isolate it from the gel. For the compounds that we're looking at, it's also very easy to determine exactly where the crosslink was, because they behave very similarly to Maxim-Gilbert G reaction. And so those of you who know about Maxim-Gilbert sequencing chemistry, our agents are hitting at the exact same site. And so the DNA falls apart at the site where you hit it, which is really very convenient. And so say we had this duplex here, and it has 2 sites for cross-linking, and I'll talk about the sites for cross-linking in a bit. But we could have a crosslink here, and we could have a crosslink here, and maybe those sites are cross-linked with different efficiencies. So we want to know how often it cross-links here and how often it cross-links with there. So again, we have a radio label here so that we can easily detect. We cross-link, we assume we have this mixture. There is probably also a lot of native and monoadduct as well, but we can isolate that through a gel. And then when we treat with piperidine, the DNA falls apart at the site of cross-linking. And so we have a short piece every time we cross-linked there, and a longer piece every time we cross-linked there. Again, we can use denaturing polyacrylamide electrophoresis to separate out those 2 products, and then we could determine the intensity of this band versus the intensity of that band and determine how often we had a crosslink at each site. So these are some of the methods that we use that I'll talk about as we actually look at some real experiments. Now, these crosslinkers have been around for a long time, and there was a lot of work in the 60s on these crosslinkers. And the investigators proposed that these crosslinkers, like nitrogen mustard and diapoxybutane, would crosslink DNA at guanines that were right next to each other, but on opposite strands at the N7 position, because this would be the minimal distance, and these are pretty short tethers with respect to the distance that needs to be cross-linked. And so here's just a little schematic here. And they furthermore proposed that the link would be at a sequence that was 5' guanine cytosine, and it's the base pairing partner to that cytosine that's cross-linked. So the 5' GC distance is the shortest, and so they predicted that that's where the crosslinks would occur. And so they actually isolated, Brooks and Lawley isolated this from DNA that had been treated with nitrogen mustard, and so you can see the chlorides left, and we have this little tether right here. And interestingly, they used paper chromatography because that's all they had back then, and that material has an RF of about 0. And so it didn't go very far, but they decided that that's what it was. When I got to the University of Washington, Professor Hopkins was interested in actually experimentally determining whether this proposal was correct. And in fact, we found that it wasn't correct. These agents, well, we were doing it for mustard at the time, crosslink guanine and a guanine on the other strand, but only at the sequence GNC. So there's an intervening base pair there. And so that distance is far too long to be linked by this fairly short tether, but DNA is conformationally flexible. And so it, in fact, does occur. We found the same thing for diapoxybutane, even though that's a shorter tether, and also for epichlorohydrin, which is a shorter tether again. So really, the DNA is not static. It has quite a bit of flexibility to it. And so it would cross-link these Gs with an intervening base pair and look something like this. But during the course of some of our studies, we noticed that there actually looked like there were some subtle differences, even though this is the core sequence preference for all of these agents. It looked like there was some subtle differences, and I'll show you why we concluded that. This was actually an image from a paper in which we were comparing nucleosomal DNA to free DNA. And so nucleosomal DNA is how the DNA is packaged, the nuclear DNA inside the nucleus. So the DNA is wrapped around this core particle of histones, and that helps package it. And you might expect that crosslinkers wouldn't be able to get in to access the DNA as well if it's tightly coiled up in these nucleosomes. And so what we were looking at was for 3 different agents, and this one is diapoxyoctane with an 8-carbon tether, the difference in the electrophoresis patterns between free and nucleosomal DNA. And the point of our paper was that the patterns are similar for each of the agents. So with nitrogen mustard, the free and nucleosomal cross-linking pattern is the same. You might be wondering why there's all these bands. These are geometric cross-linked isomers. So our piece of DNA is about 152 bases, and if you have a cross-link in the center, that has a different mobility than if you have a cross-link out on the end. And so they're similar between the pairs, so diapoxybutane again has the same pattern, free or nucleosomal, but there's some subtle differences between the agents. So if you look here, we have these dark bands here for diapoxyoctane, and they're not really there in the others. So we concluded that there's some difference in the cross-linking efficiencies at different sites, probably because of the bases flanking that consensus sequence. And so if we could understand the molecular features of these agents that dictate where they're going to cross-link, that would be very helpful in understanding how they work and possibly allow other people, not us because we don't make molecules, but other people may be able to design better anti-cancer drugs. So we systematically varied the environment of that GNC sequence to look at the cross-linking efficiencies and how they varied. And so if you have the GNC sequence, you have an intervening base pair, that could be important, and the base 5' to that G could also be important, and by symmetry that's the same as that one right there. And so we could systematically vary these and see how they affected cross-linking. And so what we did, instead of having these sequences in different duplexes, we put all 4 sequences within the same duplex and then looked at how the crosslinker partitioned amongst those. So that would minimize experimental errors between samples, which, you know, if you crosslink the same duplex, you can get different answers every time. But if you have the same duplex with all 4 possibilities and look how the crosslinker partitions, that seemed to be a much better way to design it. Okay, so for a typical experiment, what we would do is we had duplex DNA that contained all 4 sites. We had to radio label that so we could detect it with X-ray film. And then we would react with our agent of interest, and we did this same 3 family of compounds again. We could then easily purify our cross-linked DNA, and that's to show you what it looks like. If you don't have a big, long piece of DNA with lots and lots of sites, you do get a pretty narrow band here. And so this is a time course, and you see more cross-link growing in at longer times. Cut that out so that you have purified crosslink, and then treat with piperidine, and the DNA should just fall apart at the site at which it's alkylated. And then you separate via D-page those products again, and you get something that looks like this. And so these are the G's with the different N2 in this case, so that would be the base in between the N position in GNC. And so you can see, for instance, here we have a darker band here than here, and so HN2 preferred the A relative to DEB. And so we could quantitate and determine the effect of these different bases. And so we found for the 5' base that purines were preferred over pyrimidines for all 3 of the compounds that we looked at, with G being the best base and C being the worst. And for the intervening base pair, we actually found some differences, pretty dramatic differences. Diapoxybutane strongly preferred a GC. Diapoxyoctane, which is chemically similar but just a longer chain, preferred an AT, and nitrogen mustard didn't really care what the intervening base was. So we wanted to prove that this all would work. So what we did was we came up with a series of test duplexes that contained the best and worst possible combinations for DEB, and then subjected those duplexes to each of the agents. We expected there might be some difference in cross-linking. And so best, intermediate, worst, And when we subjected those to the different agents, we saw really pretty dramatic differences. So for the worst duplex, diapoxybutane really didn't cross-link it at all. And the best one, so this is just what we would have predicted, right? For nitrogen mustard, you see there's not a whole lot of difference between the cross-linking of the three duplexes. And so we could then quantitate. And we found that for diapoxybutane, there's about a 6-fold difference in cross-linking of the best and the worst duplex. And for nitrogen mustard, there really wasn't too much difference. So we felt that these findings could really explain why, when you have a long duplex with a lot of target sites with different flanking effects, why you get these differences between the different agents. So what that means is that if you have a cell with 3 billion bases of DNA, there's going to be lots of these different flanking sequences, and so we could start targeting different regions by these different agents. But what we needed to do at this point was actually look at sites that are cross-linked within cells. And so for a chemist doing cell culture, that's really biology. And so I really felt that we were going over to the dark side, having to do these experiments. I had 0 experience in cell culture, but fortunately, this is the time that I got the Dreyfus funding, which was very nice because I could hire somebody. And so I used as a model the Dreyfus fellow, the teaching research fellow, and hired Dr. Fred LaRiviere to my lab. And he came for a year, helped us get up to speed with cell culture, and then we could pass on that information down the subsequent students. He's now at Washington and Lee, so the year at Colby helped him to get that position, and he was just granted tenure, actually. So that was a great collaboration. So the way we measure reactivity in cells, or one of the ways we're doing it, is through experiments that use the polymerase chain reaction. And so one of my biologist friends laughs about this quantitative polymerase chain reaction because sometimes it seems to be pretty qualitative. It's hard to get really quantitative numbers. PCR often fails anyway, and so our experiments are actually designed to measure the amount of failure, and if it just failed, then that can be problematic. So they're a little bit frustrating to run these experiments, but they're very powerful when they work. So just to show you briefly how the polymerase chain reaction works, if you have a target DNA that you've extracted from whatever, in this case it's cells that have been treated with our nasty compounds, then you can put it into a thermocycler which heats up the strands, breaks those hydrogen bonds. You then put primers that are complementary for the regions that you want to amplify in, and so those will form little short duplex regions, and then DNA polymerase can come in and copy and make a new strand. So you started with 1 strand, you have 2 strands, and then you do this a bunch of times and you get millions of copies. And so what we can do is have specific primers for particular regions and start looking at the amount of amplification, and where the DNA has been damaged by our nasty compounds, we get less amplification. So the more damage we have, the less PCR product we end up with. And because PCR could fail, we always need to have an untreated control that we compare these to. And so we're looking at relative amplification. How well does our controlled DNA amplify, and how well does our DNA that's been treated with our compounds amplify? So when we run these experiments, we have cells that are growing up in culture, and we expose those to DNA damaging agents, so genotoxicants. We then isolate the DNA and quantify it because we have to make sure that we have the same amount of DNA in all of our PCR reactions. And then we can run the PCR with radio-labeled primers. We're now moving to try to use real-time PCR for these experiments since we got one of those 3 or 4 years ago, but we haven't quite worked that out yet. We can then separate, again using electrophoresis, our PCR products and monitor the amount of PCR product. And the higher the dose, the more damage, so the less PCR product we would expect. And then we can quantify that damage by looking at the relative amplification of our damaged over our controlled DNA. So there are first set of studies were within these chicken erythroid cells that are really easy to grow up. So that's why we started with those. They're really cell culture for dummies, which is what we felt like. And so we had 3 different loci. We had 2 that were targeting nuclear DNA. And so here's our histones right here. And if you have DNA that's being expressed, so a gene that's being transcribed, then the DNA is going to be less tightly associated with those histone core particles. It's going to be a more open structure. And if it's not expressed, it can be wrapped up very, very tightly. And so we thought maybe there would be a difference in the ability of these agents to get in and cross-link if we have a more open structure versus a more condensed structure. And then we also looked at mitochondrial DNA, which is very different than nuclear DNA. There's only about 16,000 base pairs. It's a circular DNA, and it has no packaging proteins at all. And there are multiple copies in each mitochondria. So you may have thousands of copies in a cell. It's technically much easier to amplify as well. And so we wanted to compare. Does the packaging affect it? Is there a difference in the sites targeted and so forth? And so this is what the data look like. And so here's for epichlorohydrin, and here's our mitochondrial DNA, and this is over time. And so here's our PCR product over time. And you can see that we have less PCR product over time, and we can quantitate that. But with the nuclear DNA, we have a lot less PCR product over time. So there's a real difference here in the epichlorohydrin targeting of mitochondrial DNA versus nuclear DNA. And so if we then take other data, and this is for diapoxybutane, so the data looked something like this. It was the gel images, and then we did our relative amplification. It really looks very similar for all 3 types of DNA. And so this was surprising to us that there was no difference that we could detect between expressed and unexpressed DNA, despite the packaging proteins. And mitochondrial DNA, which is naked, you'd expect it to be, or I would expect it to be, very accessible. There was absolutely no difference in how well diapoxybutane could target it. But there are some differences between the 2 different agents. And so because this one targets nuclear a little bit easier. And so our conclusions were that we saw similar reactivity by diapoxybutane at all 3 sites, but epichloro and epibromohydrin, a chemical cousin with enhanced reactivity, showed preference for nuclear DNA relative to mitochondrial DNA. And so this could just be uptake. And so the mitochondrial DNA is really quite impermeable. It's extremely nonpolar. And so it could be that these compounds just can't get inside the mitochondria as well. But this could actually help to explain cell death that's induced by these compounds, because mitochondrial DNA damage has been linked to a type of cell death called apoptosis, which is from the Greek for dropping off, like the leaves drop off in the fall time. And so the reason the leaves drop off is because some cells die, they commit cell suicide, and then the leaves can drop off. And apoptosis, here's a kind of a simplified cellular pathway. This is really a lot of biology, but it's a programmed cell death. A sequence of events occurs that is kind of a neat form of cell death in which the building blocks of the cells are recycled, and locally there's not a whole lot of damage. And so this is actually, anti-cancer drugs may work through this mechanism, and that would be very good because then you wouldn't have a lot of damage to nearby cells. And so if there's differences in mitochondrial DNA damage between these agents, there could be differences in how they induce apoptosis or not. So what we started looking at fairly recently was whether the tendency to induce apoptosis differs amongst these crosslinkers and whether that could help explain their differing biological effects. So there's a lot of different assays you can do for apoptosis, and I'll just show you one that's pretty easy to figure out how it works. And apoptosis produces this caspase 3, which runs around cleaving different proteins. And so there's an assay that you can do that monitors production of caspase 3 because you put an artificial substrate in, and when caspase 3 is present, the substrate becomes luminescent. So you look for an increase in luminescence. And so here's our luminescence here, and over time you might expect to have more luminescence, and in fact we have increased luminescence under certain conditions with diapoxybutane. So here were our conditions here, and under similar conditions epichlorohydrin does not induce apoptosis because there's no fluorescent signal produced. There's another method that I'm not going to explain, but it uses a flow cytometer, and you can actually distinguish between apoptosis and another form of cell death, which just basically degrades everything in the cell. That one's called necrosis. That's not a messy, that's a messy form of cell death rather than a nice, clean, controlled form. And therefore, you can determine the percentage of healthy versus apoptotic versus necrotic cells using this particular method. And so what you can do is with different doses kind of figure out which dose gives you the maximum amount of apoptosis and the minimum amount of necrosis, which would be much more favorable for a anti-cancer agent if you could minimize the nasty kind of cell death and maximize the clean form of cell death. And so the apoptotic potential could be the dose at which you could maximize apoptosis and minimize necrosis, and we can see that it differs for nitrogen mustard and diapoxybutane. And so this data is a senior honors student who's actually collecting this kind of data right now, and so he's done several different reaction conditions and trying to get good reproducible data. No error bars on these data yet. So we find that we can induce apoptosis with epichlorohydrin, but it takes much higher concentrations than of diapoxybutane. And we're actually we've moved now to human cells, and so this is HL60s, or a type of human leukemia cells. And we know that many anti-cancer drugs work by triggering apoptosis, and so this possibly could explain why diapoxybutane, or partially explain why diapoxybutane has anti-cancer activity and epichlorohydrin does not. We're also interested in the mechanism of apoptosis. It's a very complicated pathway, and so there are two major ways you can form or have apoptosis developed, an extrinsic pathway and an intrinsic pathway. And so this, again, is pretty simplified, but we have lots and lots of proteins that are produced during apoptosis. And so I have another student who's been looking at upregulation of the proteins in apoptosis to see which proteins are upregulated when we treat with these compounds under conditions that we know causes apoptosis. And so I'm not going to go into much detail here, but she's using real-time PCR to determine the amount of messenger RNA produced by these cells. And basically, I'm not going to explain these experiments, but she can start to determine which cells, which proteins are being produced, and therefore we can zone in on the actual mechanism of apoptosis. And so here's some proteins that she's found have been upregulated in the presence of diapoxybutane, and so we can say that, in fact, that our data are consistent with the intrinsic pathway. Interestingly, these proteins are not upregulated with epichlorohydrin, so there's a difference in the response of the cell to these different agents. Okay, and so that's it. It's just kind of a snapshot of things that have been going on. And these are, we've had lots of other projects over the years. These are all the students who've been involved in this research and our kind funding organizations. So thank you very much.
This is Herman Sintim from University of Maryland. So it turns out that RNA does a lot of beautiful things in the cell, but is often ignored. And I was just wondering about whether some of these anti-cancer agents actually cross-link RNA, and maybe some of the biological effects are actually due to ribosomal RNA being inhibited, and some of the sigma factors or the housekeeping genes that would regulate apoptosis are also affected.
I'll bet they are, yeah. So we haven't studied that at all, but clearly these agents are reacting with a lot of targets. For many of these agents, the amount of DNA cross-linking has been correlated with the cytotoxicity, and so that's why they think the interstrand cross-linking is the most lethal event. But I'll bet you're right. I'll bet there's a lot of RNAs that are being cross-linked as well, or damaged because you could monoalkylate. So we haven't looked at that at all, but I'll bet that that's going on.
So I just had a question about the last set of experiments that you showed. And so the first one is, are these proteins that are being upregulated when you expose the cells to the chemicals, are these proteins, I assume by the RT-PCR you're looking at messenger RNA levels?
Yes.
Do you know if those messages are actually being expressed? Namely, are the proteins themselves being upregulated, production of those proteins? And then the other question is, are these under the control of similar transcription factors?
I believe that they're not, because there's dozens of these proteins that we've looked at. So I don't know what transcription factors are controlling them, but it would be unlikely that they would all be under the control of the same ones. And so your other question was, oh, right, so are the proteins increasing? We've done some Western blotting as well, and there's some actual kits that you can get that detect actual protein levels. But unfortunately, those kits don't always coincide with the ones that you can get the real-time PCR primers for. So we have done some of those, but we haven't really intersected yet or looked at the data. So the same student who's doing the real-time has looked at, I think we got a library of like 96 primers that are involved in apoptosis, so a lot more proteins than we see up here. And she also got this kit that looked at, I think it's about 40 maybe actual protein levels. But yeah, that's a good question. Just because the genes are upregulated, are the proteins being expressed? Most people in the field tend to say that if the messenger RNA is up, then the proteins are as well. But we felt that we should follow up with other experiments.
What's known about mechanism of uptake and metabolism inside the cell, and could the differences you're looking at be related to that? One's charged, one's not charged. Right,
and so I haven't actually seen much about that, so I haven't seen much literature about uptake, but we felt that the epichlorohydrin, just looking at the characteristics of the molecule, would probably not be taken up into mitochondria as well. But I haven't seen a whole lot. There's a lot on metabolism of butadiene, and so differences in different species and so forth, but I haven't seen a whole lot of just the compounds being taken up in the literature.
Carlos Gutierrez from California State University, Los Angeles. Depending on the tether between any two epoxides, can you build linkers that might do intrastrand cross-linking?
I've seen some work where people have tried to design these new agents to do sort of similar things, but intra, it's not thought has the same lethal effect. And so there may be some structural perturbations, but the inter, because you cannot express the information, are thought to be more deadly. But I know a lot of people are trying to design agents changing tethers and changing reactive groups. We don't build molecules, but I know that there is some interest in that.